Hi everyone, my name is Matt Hurst and I'm in the chemistry department at Cal Poly Humboldt. I've been asked to give an explanation about the chemistry in your eutrophication experiment and how it mimics what we see in nature. So what you see in the slide is a cyanobacteria bloom in Klamath Lake a decade ago. And what I'll describe to you is how these blooms take place and what the requirements are on a chemical level for these blooms to occur. And so the cell division cannot happen without DNA replication. Now for cyanobacteria, a single cell of a cyanobacteria will double itself 2 times a day. So in the morning you might have 1 cell, but by the end of the day, if it has everything it needs, that 1 cell will turn into 4 cells. So when we think about the replication of the DNA, it's made up of all these nucleic acids that come together to form the strand that we've seen in the textbooks. If we look a little bit closer at DNA, what we see is that it is made up of a lot of these hydrogen bonds that contain nitrogen. So DNA, in order to produce and replicate DNA, you need a lot of nitrogen. Also, if we look at the strand, we see that the backbone on both sides is made up of phosphorus. So phosphorus is also important in cell replication. So we need both nitrogen and phosphorus to replicate cells, along with other elements that are required in the cell. So let's just take a step back and think about photosynthesis and the biological requirements and pH. So a simplified equation for photosynthesis is shown on the slide. And you all know this equation well. And so we have CO2 with water creating carbohydrates or sugars. I call it biomass, the bulk of the cell, and we release oxygen. The opposite direction for this reaction is respiration. But plants and cyanobacteria need more than just carbon, water, and light. So perhaps this equation isn't the best representation for you to think about in terms of your experiment and how photosynthesis occurs. So if you were to take a cyanobacteria cell, or a whole bunch of it, and crush them up, and then analyze what elements are in that goo that you've created, you would see that the ratio of carbon to hydrogen to oxygen to nitrogen to phosphorus would be 106, 263, 110, 16 to 1. So this is the ratio of the elements that you would see in that cyanobacteria mass. And if we put that into a chemical formula, we could say that cyanobacteria have this chemical formula, which is made up of these elements and at this atomic ratio. So if we want to really look at photosynthesis as it occurred in your eutrophication experiment, we would write an equation like this. We would involve nitrogen as nitrate, phosphorus as phosphate, and this would be a better representation of photosynthesis in your experiment. We saw that nitrogen and phosphorus were necessary for things to grow. Also, when we look at this equation now, we see that during photosynthesis, hydrogen ion or acid is consumed. CO2 acts like an acid in water. And so when we remove CO2 during photosynthesis, it will change the pH. We should see in the afternoon an increase in the pH. In the morning, after respiration occurs all night, we typically see the pH drop in the morning. So depending on when you measured your pH, you might have seen a low pH in the morning and a higher pH in the afternoon. So when we consider the entire experiment, we amended our experiment with nitrogen and phosphorus, but there are lots of other elements that are required by cyanobacteria. And in future experiments, we might want to consider adding some of these to see how they affect the growth of cyanobacteria in Klamath River water. So now let's consider the class data. You had 3 different setups, and what we saw in each setup is that by adding nitrogen and phosphorus to the samples, there was enhanced growth, more algae in those samples compared to the control. Now I will say that is just the chemistry part of this experiment. You guys introduced a lot of physical conditions that had some effect on the growth as well. So there's many things to tease out in this data, and it's not just about nitrogen and phosphorus. So last, what you guys looked into was the effects of photosynthesis on the pH of the water. And so you guys measured the pH in the morning and in the afternoon, somewhat in a broad range of time, but nonetheless, your data still shows that in the morning, the pH is lower due to the fact that there's more CO2 in the water. That CO2 is from respiration that has occurred by the organisms all evening long. Photosynthesis kicks in after 1 p.m. and especially 4 or 5 o'clock in the evening. And what we see there is a drawdown of the CO2 and an increase in the pH. So there's less H plus or acid in the water due to photosynthesis, because that's also required to make the stoichiometry work, as we discussed before. So nonetheless, I thank you guys for your work. It looks great, and I wish you the best of luck in the school year ahead. Take care.